Hey everybody, in this video I am going to provide a little bit of background on Neurospora, the fungus Neurospora, for the Hammond Lab research assistants. So Neurospora crassa is the fungus we study in my laboratory. And okay, let's just a little bit of background here on this fungus. So as I said, it is a fungus, and it's going to be more like the fungus that grows on, let's say, bread in your cupboard or pantry or something. Then let's say this is a piece of bread here. And sometimes you might see a bluish green fungus, and sometimes you might see an orange like fungus growing on the bread. Well, if it's orange, well, there's a chance that it could be Neurospora. So again, more like the fungus that you would probably see growing on bread than, let's say, a mushroom that you might see growing out in, like, on a hike or something like that. So mushrooms tend to be basidiomycetes, and the fungus we study is classified as an ascomycete. So again, Neurospora is a genus of fungi. Now, we study it because many years ago, almost a decade ago, a few researchers decided it would be a good model to understand eukaryotic genetics. So that's why we study it today in my lab and in many other labs around the world. So it is found all over the world in temperate, subtropic, and tropic climates. So far hasn't been found in polar climates or subpolar climates, but it has been isolated from these various climates. And the rumor is it is really easy to isolate it from burnt vegetation. So for some reason, after a grass fire or forest fire, and you have dead plant tissue, well, Neurospora really likes to grow on that tissue, and that's a great time to isolate it from the wild. So because Neurospora has been studied for so long by so many different people, we have isolates from around the world, and there's one particular group that really collected most of the isolates that are readily available to those studying Neurospora. So let's take a look. Let's take a look at where people have isolated this fungus from. So I have a little map here that is pre-diagrammed. And I'm referencing a study here by Turner, Perkins, and Fairfield that was published in 2001, and it is a summary of a 3-decade study where strains or isolates of Neurospora, various species of Neurospora, were isolated from various locations around the world. Now, they say they've collected over those 3 decades 4,600 cultures from 735 sites. Wow, so that's amazing, right? Okay, and here's this nice little map here, and this is supposed to be a map of the Earth, right? The various continents and places like that. And so where would you think you would find Neurospora? So we'll Okay, whoa. So that's a lot of sites. That's a lot of locations from around the world. And you can see, well, oh, okay, like we said, tropic, subtropic, and temperate. And what Turner and Perkins say, well, it's this highest latitude spot right here. Well, it isn't that they didn't try to isolate from above those latitudes. It's just that they didn't have any success isolating from above those latitudes. And we have some big gaps in here. And what they also said was that if there's a gap, it could be that the gap exists because they didn't try to isolate anything from there. So point is, Neurospora seems to be essentially everywhere except for very cold environments. So in that collection of 47 or 4,600 over 4,600 cultures, what we have are 5 defined species, and I think there are more now. I think the classification of different species has become a little more sophisticated, but the 5 main species in that collection are as follows: Neurospora crassa, Neurospora intermedia, Neurospora setophila, Neurospora discreta, and Neurospora tetrasperma. Now, Neurospora crassa, this is the genetic model. It's the one most laboratories study if they are focusing on some aspect of understanding eukaryotic genetics, particularly molecular genetics. And intermedia is so closely related to Neurospora crassa that it's somewhat difficult to actually determine or classify these as separate species. But historically, they have been classified as separate species, so I will consider them to be separate species here, but it's very closely related. So closely related, in fact, that it will be, researchers have succeeded in producing offspring from crosses between individual strains of N. crassa and individual strains of N. intermedia. Now, these 5 species, I should clarify that these are all the conidia producing species of Neurospora. And I'm going to talk about what conidia are in a moment. Actually, right now. I'm going to talk about what conidia are right now. So, now when we're talking about conidia, well, it helps to talk about the various spore types. In Neurospora fungi, or Neurospora like Neurospora crassa. So we can classify spores, so spores being propagules of the fungus, and these propagules are meant to spread to new locations. So we have asexual spores and sexual spores. Of the asexual spores, asexual spores, I should say, are produced by mitosis. And because they are produced by mitosis, they are all genetically identical. So each of these spores is going to be genetically identical, barring any mutations, that might occur during DNA replication. All of the asexually produced spores from a particular strain of Neurospora should be genetically identical. And Neurospora crassa has 2 main types of asexual spores. Main types, I should just say types. I don't know of any more than 2. I only know of 2. So macroconidia and microconidia. Now, conidia is the plural form of the word. We would say conidium. So macroconidium is the singular, and microconidium is the singular of this form over here. So macroconidia have an average diameter of about 7 micrometers, and microconidia have an average diameter of about 3 microns or micrometers. So these macroconidia contain an orange pigment. So for those of you who are already working in my lab, you know that Neurospora cultures are typically orange, and that's the orange pigment that's found in the conidia. Now, microconidia are produced in much lower levels than macroconidia, much lower levels. And we actually don't talk much about microconidia. To get large numbers of them, or enough of them to notice them, you need to be the fungus needs to be grown on a special medium. So most of what we work on is going to be work with, I should say, are macroconidia. So what about the sexual spores? So these, you can probably guess from their name, sexual spores. They are produced during mating. They are produced specifically by meiosis or after meiosis. They descend from a meiotic event. So without meiosis, they cannot be produced. And because meiosis produces them, they are genetically different. They tend to be genetically different, right? So each ascospore, each sexual spore, I should say, is going to, well, let me point that out first. They are called ascospores. So the sexual spores are called ascospores. And half of their genetic content is from parent one of the parents, right? The fungus is going to have 2 parents in a mating event. So half of the DNA is from parent 1, half of the genetic content, I should say, is from parent 1. The other half is from parent 2. Now, ascospores look like American footballs, and they are darkly pigmented with a dark brown pigment, so much of it that they appear black. And they are somewhat large relative to, let's say, the macroconidia. So a typical ascospore is 14 microns wide and 28 microns long. Okay, those are the ascospores. Those are the sexual spores. They are produced by meiosis, or at least they require meiosis to be produced, and we'll learn more about how they are produced in a moment. So some other things I want to touch on with respect to especially the asexual spores, the macro and microconidia. So asexual spores, as I said, we have macroconidia and microconidia. Now, in the lab, we almost never differentiate between macroconidia and microconidia. When we say today we are going to be isolating conidia for transformation, we're always talking about macroconidia. So we almost never use the word macro, almost never. We always just call them conidia. If we are ever referring to microconidia, well, we will say microconidia. So in the lab, when we say conidia, we mean macroconidia. Again, the singular form is going to be conidium, and the singular form of microconidia is going to be microconidium in the lab. Now, those of you who've already started working in the lab, you probably cultured the fungus in these little flasks, and a small amount of solid media at the bottom, and the fungus grows up here, and you get all these orange conidia up here. Well, yep, those are macroconidia. Okay, so let's now take a look at the life cycle of N. crassa. So again, focusing on Neurospora crassa, and the life cycles of the other species are going to be very similar to that of Neurospora crassa. So let's start with a conidium. So one conidium diameter, average diameter 7 microns. So let's say the conidium finds itself in a suitable environment for growth. What will happen is it will germinate. And we'll have this little germling here. And fungi like Neurospora grow, display apical growth. And what does that mean? So essentially it means it's gathering nutrients from its environment, and it's making the cellular components it needs to grow, and it's transporting them up towards the tip here. So it's the tip that grows. The fungus grows from the tip. So as that grows, it will tend to branch, and the branches also grow from the tip. And what we can do is we can call these various segments here, we can call these hyphae, which is the plural of a hypha. So hyphae would be multiple of these tube-like structures. Hypha would be like one tube-like structure. If I were to like close that off, like block off that branch right there, that would be a hypha. So Neurospora divides these into compartments that are separated by septa. So septa, these like cell wall type structures that come out and kind of divide the hyphae into individual compartments. Now, in Neurospora, most of these septa have pores. We can call these septal pores. And what that means is the cytoplasm can go through the pores. And these aren't individual cells. They're not individual cells. They're not separated from each other. They share a cytoplasm. And not only do they share cytoplasm because of those pores, they also share nuclei and mitochondria and anything that you can think would be inside the cell. So nuclei can flow through the pores also. And each compartment can have dozens of nuclei. Okay, that's crazy to think, right? Because usually we're thinking of animal cells and plant cells, where typically we're thinking there's one nucleus, right? One nucleus usually per cell. Oh, here we're going to have dozens of nuclei in each compartment. And those compartments tend to be about 15 to 40 microns. 15 to 40 microns. Now, you'll see another word in the lab, mycelia. So when we say mycelia, essentially what we're talking about is a mass of fungal hyphae. And usually when we're using the word mycelia, we're talking about a part of a culture that does not include the conidia or other spore types. Mycelia, just a mass or collection of hyphae. Okay, so now that we know a little bit of how a Neurospora culture gets started, let's say from a single conidium, what I want to do is talk a little bit about the nuclei. So, as I mentioned, Neurospora crassa forms these compartments that aren't really separated from each other. Now, those pores can be plugged up, I think, if the fungus if a hyphal segment becomes damaged. But for the most part, the hyphal compartments are connected, and the cytoplasm of the nuclei can go back and forth between the various compartments. And so let's assume dozens of nuclei per compartment, depending on the age of the hyphal segment, developmental stage of the hyphal segment. Now, with respect to a single nucleus, let's take a look at the nuclear DNA. So inside a single nucleus, we will have 40 million base pairs of DNA spread over a single set of chromosomes. And in a single set, there are 7 chromosomes. Now, why do I say single set? Because nearly all of the nuclei in N crassa are haploid. So if you consider, if you're thinking of a Neurospora crassa nucleus, 99.999% of the time it's going to be a haploid nucleus. There's just one nucleus during meiosis, at the very beginning of meiosis. One nucleus at the beginning of meiosis actually is diploid. And we're going to talk about that in a moment. So nearly all of the nuclei are haploid, but at the very beginning of meiosis, we're going to have 2 haploid nuclei fuse to make a diploid nucleus. And then that's going to quickly break down into 2 haploid nuclei again. Okay, what does haploid mean, right? Just checking. One set of chromosomes. So a haploid nucleus has only one set of chromosomes. And like I said, a complete set of chromosomes in N crassa is 7 chromosomes. So just briefly, because this is going to be important for one of our main projects in the laboratory, is knowing a little bit about each chromosome. And so let me diagram these chromosomes here. And when I diagram a chromosome like this, for those of you who have taken genetics with me, this is a chromosome before DNA replication, right? So 1, 2, 3, 4, 5, 6. These are 7 chromosomes before DNA replication. So if a chromosome undergoes DNA replication, they look like that. So this is a single chromosome with a pair of sister chromatids. So here I've just diagrammed them as single chromosomes before each containing a single chromatid, 7 chromosomes before DNA replication. And so if this is chromosome 1, here's 2, 3, 4, 5, 6, 7. So remember, Neurospora has 7 chromosomes, and they have numbers. 1, 2, 3, 4, 5, 6, 7. So number 1 here is 10 million base pairs. Number 2 is about 5 million. Number 3 is about 5 million. 4 and 5 are each 6 million base pairs, and 6 and 4 are each 4 million base pairs. Now, number 3 is going to be very important for those of you studying spore killer, at least spore killers called spore killer 2 and spore killer 3 in my laboratory. So now I mentioned up here about this one type of nucleus which becomes diploid at the very beginning of meiosis. And meiosis occurs after mating. So Neurospora crassa mates to produce offspring. So a little bit about mating in Neurospora crassa. So Neurospora crassa mating. So mating occurs only between opposite mating types. I hope I'm still on camera here. I think so. Yeah. Mating occurs only between opposite mating types. And there are, guess how many? Only 2 mating types in Neurospora crassa. Only 2 mating types. Unfortunately, they have names like this: big A and small a. So one mating type is called big A, the other mating type is called small a. So when we're talking to each other in the laboratory, we'll say, what's the mating type of that strain? And someone will say, oh, it's big A. And someone else might say, no, that is small a. So yeah, that is how we have to describe the mating types. That's how they're defined. So it can be a little confusing sometimes. And okay, so what determines a mating type? So let's say this is chromosome 1 in a random strain of Neurospora in a nucleus. And chromosome 1 has a locus called the mating type locus, or the MAT locus, MAT for short. So the genes at the mating type locus determine whether a strain is the big A mating type or the small a mating type. So we could take any strain, we could use some primers and do some PCR and some sequencing and determine just by the sequence of the DNA at the mating type locus whether that strain is a big A strain or a small a strain. And again, the mating types are important because mating only occurs between opposite mating types. If you try to get two strains of the big A mating type to mate, they wouldn't mate. They would not mate. Same thing for the two strains of the small a mating type. So, okay, how does mating work? And crassa mating. So we know two mating types exist. Now, let's just take any old strain, could be either mating type. So under appropriate environmental conditions, that strain will make what we call protoparesthesia. That's the plural. The singular is called a protoparesthesium. And so it will look something like this. And this is remarkable, and we know very little, even though we've been studying this fungus for hundreds of years, we know very little about the developmental control of the formation of a protoparesthesium. So somehow those fungal hyphae, instead of growing to make more hyphae, they start growing into a protoparesthesium. Now, the protoparesthesium is an immature fruiting body. And so what will happen during mating is either, well, let's just say this little structure here, which looks like a hyphal segment here, it is a trichogyne, trichogyne is hair-like. So this is a trichogyne, which is a hair-like structure that grows out from the protoparesthesium. Now, either it grows out before or it grows out of the paresthesium when it senses some sort of tissue from a strain of the opposite mating type. So let's say this is the protoparesthesium from a strain of the small a mating type. Let's say this is a conidium of a strain of the big A mating type. So the trichogyne might develop when this is in the vicinity of the protoparesthesium, or it might start developing, hoping that it comes close within the vicinity of conidium of opposite mating type, or other tissue type of opposite mating type. And so there is thought to be some chemical signaling involved here, where the trichogyne will grow towards the tissue of opposite mating type based on the chemical, based on the density of the chemicals in the environment. And what will happen is we'll get cellular fusion between these two tissue types here, and a nucleus from, in this case, the conidium will travel down the trichogyne into the protoparesthesium, which is now called a paresthesium. So after that nucleus, and I didn't talk about this before, but let's say a macroconidium here can have 3 to 8 nuclei in there. One of these will travel down into the protoparesthesium, and after that happens, it's defined as a paresthesium. It's an immature paresthesium. But after fertilization, the protoparesthesium is considered a paresthesium. Okay, now that's going to continue to develop. It's going to continue to develop in there, and that's where the offspring, the meiosis is going to occur, and the offspring or the ascospores will be produced. Okay, so within the protoparathysium, within the parathysium after fertilization, inside the developing parathysium, the hyphae are going to be dikaryotic. Now you can use dikaryotic in a couple of different ways. You could say a cell is dikaryotic because it has 2 nuclei. Here I'm using it to mean we have nuclei of different mating types in here, right? We've got the small a from one parent and the big A from the other parent. And as I said before, the hyphal compartments can have many nuclei. And so here we have many of each type of nucleus in the various compartments. And so the hyphae inside the developing parathysium are going to have many copies, many nuclei, some from one parent, some derived from one parent, others derived from the other parent. So some of the big A, some of the small a mating type. And again, this is poorly understood, but what is known is that inside the developing parathysium, these hyphae with both of the parental nuclei in there will give way, or what will be produced from these hyphae here are these Crozier hook cells. Crozier hook cells. So Crozier is like a hook. It's a hook. Crozier hook cells. And what we have in the Crozier hook is we have the various nuclei of the big A and the small a type, and at the very end of the Crozier hook, there is going to be what we're calling the pentultimate compartment here. The pentultimate compartment. Let's say this is the last compartment. This is the pentultimate compartment. We'll have one of each nucleus. And so it's really remarkable how the fungus does this. No one really understands all of the controls or genetic control and of the developmental processes that occur to make this happen. But what has to happen is in the Crozier hook, we need to have one nucleus of the big A type, one nucleus of the small a type. In this pentultimate cell, with these 2 haploid nuclei at this point, is going to become the meiotic cell. And that's going to happen after these fuse, after these nuclei fuse. So I have some diagrams already made for us to speed this process up. Okay, so here's that little Crozier hook. Now I'm calling this pentultimate cell here with the 2 haploid nuclei, the proto-meiotic cell. So those 2 haploid nuclei will fuse to form a diploid cell. Okay, what does that mean, right? Okay, each haploid nucleus has 7 chromosomes, right? And let's say each of those chromosomes underwent DNA replication. So now they're represented as they're represented as pairs of sister chromatids. When they fuse, now we have 14 chromosomes in there. We have 2 sets of chromosomes, 2 sets of 7 for 14 total. That's the diploid nucleus. So once those cells form, we're calling this last cell here, we're calling that the meiotic cell. And it will elongate. It will elongate to look something like this. So what is it doing when it's elongating? It's forming what we are going to call the ascus or spore sac. So this is this elongated cell. And often we'll consider meiotic cells or a meiotic cell and the ascus to be the same thing. The plural of ascus is asci. So after the diploid nucleus is formed, well then the stages, the typical stages of meiosis occur. We'll talk more exactly about how the chromosomes are behaving during these various stages, probably in a later video. But here, okay, meiosis 1 is occurring. So maybe you remember from genetics that meiosis is divided into 2 stages, meiosis 1 and meiosis 2. So meiosis 1 is the reductional meiotic division stage, where we decrease the number of sets of chromosomes. So we go from 2 sets here to 1 set here. So meiosis takes this diploid nucleus, and after meiosis 1, we have 2 haploid daughter nuclei, and we're going to stop paying attention to the mating types at this point. The next stage is meiosis 2. So these are each here undergo the second meiotic division, these nuclei here. Now nothing's happening really. The cell might be getting bigger. The ascus of the meiotic cell might be getting bigger, but nothing much is changing with the cell, right? The cell's not dividing, just the nucleus, just the nuclei are dividing during the various meiotic stages. So these nuclei are going to go through meiosis II. So this one is going to make 2 daughter cells here, and this one right here is going to make these 2 daughter cells here. And these are also all haploid. These are also all haploid. And that might be a little confusing without the diagram of the chromosomes. But essentially, each of the 7 chromosomes here looked like that. They're called dyads, right? And now at this stage here, they look like monads. Okay, so that's why we're going from haploid here, 2 haploid cells here, through meiosis II to 4 haploid, not cells, sorry, 4 haploid nuclei here. So 2 haploid nuclei here, they go through meiosis II to give us 4 haploid nuclei here. Now, Neurospora is kind of weird. Normally when we think of meiosis, we think of 4 meiotic products, right? So let's say spermatogenesis in animals. 1 meiotic cell will make 4 sperm, 4 meiotic products. Well, here in Neurospora, what happens, each of these nuclei here undergoes mitosis. So now the ascus contains 8 nuclei. So 1, 2, 3, 4, 5, 6, 7, 8. 8 nuclei. And we can consider each pair to have resulted from mitotic event from a product of meiosis. So these would be genetically identical here. Like these 2 are genetically identical, these 2 are genetically identical, and so on and so forth. Now that we have 8 nucleus in here, 8 nuclei in an ascus, what we have occur is ascosporogenesis. So the ascospores, the sexual spores, start to develop. 1 ascospore around each nucleus. So I have that diagrammed here. So you can see each nucleus here, and you can see these ascospores, they're sort of being cut. We're getting cell walls and cell membranes built around each nucleus. They're being cut from the surrounding cytoplasm in that ascus. Now, as the ascus is developing, the ascospores will become darkly pigmented. And I believe it's melanin. And melanin helps protect the spores from UV light and harsh environments. And so this is what a mature ascus is expected to look like. So a single ascus, a single spore sac derived from a single protomeiotic cell should have 1, 2, 3, 4, 5, 6, 7, 8. 8 ascospores. These ascospores are all siblings, and these 8 are all derived from the same meiotic event. I want to touch on the plural of ascus is asci. Now, we talked about this already. Here's each of these is a sexual spore, ascospore, about 28.5 microns long versus 14 microns wide. Now, if we were to look inside a perithecium, right, we're going to get way more than 1 ascus. We're going to get something that looks like this. Okay, and I have the asci at this particular stage in this perithecium having ascospores of different levels of pigmentation. That's because the asci develop asynchronously. Oh, sorry, it's been a long day. Spelling is terrible right now. Asynchronously, I guess that's close enough. Asynchronously. They develop asynchronously in the perithecium. So some of the asci in the perithecium will be further along in development than others. So some may have 8 darkly pigmented spores. Others will have more lightly pigmented spores. But if we waited long enough, they should all have dark black appearing spores. So I just use these various colors here to bring up the point that the asci develop asynchronously. in Neurospora crassa parathecia. So a parathecium is about 400 to 500 microns long. That's what 500 microns. That's pretty easy. We're getting up to levels that we're used to seeing, right? Is essentially half a millimeter. So they're pretty big, and we can have 200 to 400 asci inside a parathecium. So that's a ton. And what you'll all probably be doing in the laboratory is dissecting. One thing I'll touch on, which I'm not going to talk more about in this video, but in later videos I will, is when they reach maturity. When they reach maturity, they can be shot. Each ascus is shot from the parathecium. It's pretty neat how that process works, but they're shot from the parathecium. And what we like to do in the laboratory is it's much better to study these asci before they're shot. And so what we'll do is we'll dissect with needles. We'll pull these parathecia off of the crossing plates, and we'll dissect them in like 25% glycerol to visualize the developing asci before they're shot out of the parathecium. And if we're careful, we should see something that looks like this. So this is after all the parathecial wall fragments are separated. And I'm just going to put a couple of wall fragments here. And there might be some other tissues in here also. This is a rosette of asci, and I didn't diagram all 200 of them, but I've just given you an example. And they tend to come out of the parathecium in a rosette, almost like a rose with the petals, emanating from a central location. And so here's a rosette of asci, and we can make out the borders, right, of each ascus here. And it's hard to see the cell walls of asci because they're translucent, but they hold the spores together. And so we can see, oh great, okay, each of these asci, each ascus is the result of a single meiotic event. And we like to do our section somewhere at right about 2 weeks. Right about 2 weeks after fertilization, after that fertilization of the protoparathecium occurs. You could go as long as 4 weeks, but then, by 4 weeks, I think most of the asci will probably have been shot out of a parathecium. So, okay, so now, what is all that? Okay, so now, I mentioned that earlier study from 2001, which was Turner and Perkins and Fairchild or Fairfield, I forget exactly, but sorry for the third author. So over 4,600 strains from hundreds of different locations, and when they were examining these strains, and they were performing crosses and dissecting parathecia to determine, what species each of this isolate, which species of Neurospora each of these isolates belong to. Once in a while, they noticed something peculiar with respect to the rosettes. And so, like I said, usually these rosettes of asci contain asci with 8 black ascospores. Okay, that's normal. Once in a while, they would see asci with 4 black viable ascospores like normal, and 4 shriveled up hyaline. Hyaline means translucent spores. And so the 4 black ascospores, which were normal sized, were viable, and these 4 shriveled ones were dead. They were dead. And so each ascus in the rosette would have that same phenotype: 4 normal looking ascospores and 4 dead ascospores. And what they have discovered was spore killing. So they discovered spore killing. And this was 1979 when they first reported this. Remember that 2001 was a summary of 3 decades of collection and study of these strains. Well, 1979, it's in that 3 decades. So in 1979, they reported this phenomenon. And what they had discovered was 3 spore killing strains called SK1, SK2, and SK3. And my lab really focuses in on these 2 right here. And I think this semester, I think what most of the undergraduates are studying right now is what we're going to call SK3. So again, discovered in 1979, very little is understood about spore killing, and we're trying to figure out how it works in my laboratory. Okay, so I think in the next video, I'm going to take a look at this paper specifically from 1979 when they discovered spore killing. See you in the next video.